Hello viewers, I am Dr. Rabiul. I work as a lecturer of pathology in a medical college hospital and I am making this video for my students and also for you. Hope someone finds this helpful. Today's topic is on type I hypersensitivity. In this lecture, we will learn about the definition of hypersensitivity, its general features, classification, immunological aspects, and then we will mainly focus on type I hypersensitivity and discuss the sequence of events that occur during this type I hypersensitivity, the mediators involved in this process, the clinical aspects, and towards the end of today's lecture, we will also talk briefly regarding the late phase reaction. Okay, a lot of topics, so let's begin. First question, how can we define hypersensitivity? It may be defined as exaggerated or inappropriate immune responses to benign antigens. Okay, I am repeating this definition again for my students and then I will explain this definition. Hypersensitivity may be defined as exaggerated or inappropriate immune responses to benign antigens. Now, what do we mean by this term, benign antigen? This is in fact referring to certain exogenous and endogenous antigens that are normally harmless. For example, exogenous antigens like antigens that are present in dust, pollen, drugs, foods, microbes, chemicals. Normally, these harmless antigens will not result in hypersensitivity reaction. However, in certain individuals, we will see that these harmless exogenous antigens may cause inappropriate or exaggerated immune response, and those inappropriate or exaggerated immune responses are referred as hypersensitivity. So we can say that hypersensitivity is an excessive or harmful immune reaction to antigen. So now that we have defined hypersensitivity, now let's move on and talk about the general features. We have already talked about the first point. Hypersensitivity reaction can be evoked by exogenous environmental antigens or by endogenous self-antigens. Examples of exogenous antigens will include antigens that are present in dust, pollen, drugs, foods, microbes, or chemicals. And always remember, immune response may also occur against endogenous self-antigens, and that is also referred as autoimmune disease, and we will talk about that in details in another lecture on autoimmune disease. Regarding the general features of hypersensitivity, always remember it usually results from imbalance between effector mechanism and control mechanism of our immune responses. And again, it is often associated with inheriting certain susceptibility genes. That's why some individuals will be hypersensitive while others will not be hypersensitive to certain antigens. And as we can see, the basic mechanism is poorly controlled, excessive, or misdirected immune response against normally harmless environmental and self-antigens. Now, this bullet point is very important. Always remember, it is the immune response, not the antigens, that are harmful to the host. Again, hypersensitivity is antigen specific. First contact with the antigen will sensitize or prime the individual, and upon subsequent contact, there may be hypersensitivity reaction. And as we will see, severity may increase with time. So now that we have talked about the general features, now let's move on and talk about the classification of hypersensitivity. It is classified on the basis of immunological mechanism. But always remember, multiple mechanisms may be involved in any one hypersensitivity disease. So it is classified in 4 types. They are type I or immediate hypersensitivity, type 2 or antibody-mediated hypersensitivity, type 3 or immune complex-mediated hypersensitivity, and Type 4 is also known as cell-mediated or delayed type hypersensitivity. So now that we have classified hypersensitivity, now let's talk about the immunological aspects of hypersensitivity. What is happening in type 1? Here, antigens will cross-link immunoglobulin E antibodies that are present on the surface of mast cell. Now note that in this slide we can see a term allergen. Now always remember the antigens that can cause type 1 hypersensitivity reaction are often referred as allergen. So that's why I had written allergen in this slide. So what is happening here? First, allergens will cross-link immunoglobulin E antibodies that were present on the surface of mast cell. That will result in degranulation of the contents of mast cell, and that will release mediators and result in hypersensitivity reaction. What will happen in type 2 hypersensitivity? Antibody will bind to cell membrane antigen and it will cause complement-mediated lysis of the cells. What will happen in type 3 hypersensitivity? Here, antibodies will bind antigens usually in the circulation. So there will be formation of antigen-antibody complex in the circulation, and then that complex will be deposited in tissues and that will induce inflammation. What will happen in type 4? Here, sensitized T lymphocytes will cause tissue injury. So now let's focus our attention on type 1 hypersensitivity, and throughout the rest of today's lecture we will mainly talk about type 1 hypersensitivity. So type 1 hypersensitivity is also known as immediate hypersensitivity, and as the name implies, it is a rapid immunological reaction. The reactions are often called allergy, and the antigens that can cause type 1 hypersensitivity are often referred as allergens. It occurs in a previously sensitized individual, and as we have already mentioned, it is triggered by binding of an antigen or allergen to immunoglobulin E antibody on mast cell surface. And always remember, it may occur as a local reaction or as systemic disorder. Now, most immediate hypersensitivity reactions will occur due to excessive T helper 2 response, and that will occur in the following sequences of events. First, T helper 2 cell activation and production of immunoglobulin E antibodies will occur, and as we will see later, sensitization and activation of mast cells will occur. So let's talk about these various sequence of events one by one. So now let's look at an animation that I have made using the software Blender to show you the sequences of events of type 1 hypersensitivity. First, let's talk about T helper 2 cell activation and production of immunoglobulin E antibodies. So what is happening? First, we can see an antigen that has entered into our body. What will happen next? Antigen presenting cells will try to uptake that antigen. Now, whenever you mention the term antigen presenting cells, the examiners are very fond of asking you what are the antigen presenting cells. And always remember, macrophages, dendritic cells, and B lymphocytes, these are the antigen presenting cell. So here in this animation we can see a dendritic cell is now taking up that antigen. What will happen next? After uptake of the antigen, the dendritic cell will process the antigen and it will present the antigen on its surface with the help of MHC class II molecule. Recall that the antigen was extracellular, that's why it is expressed with MHC class II molecule. What will happen next? Now the dendritic cell will take that antigen and present that to naive T cell or CD4 positive T cell. In response to antigens and also due to some locally produced cytokines like interleukin 4, now this naive CD4 positive T cell will differentiate into T helper 2 cell. And always remember, T helper 2 cells will begin to produce interleukin 4, interleukin 13, and interleukin 5. Particularly, interleukin 4 is very important because, as we will see, interleukin 4 is required for immunoglobulin E class switching. So what do we mean by that, immunoglobulin E class switching? In order to understand that, now let's look at the animation, and now we can see that the same antigen is now presented to the T helper 2 cell, but this time not by dendritic cell but by B lymphocyte. Recall that we had said 3 cells are in the category of antigen presenting cells, and they were dendritic cells, macrophages, and B lymphocytes. So now B lymphocytes are presenting the same antigen to T helper 2 cell, and since T helper 2 cell is producing interleukin 4, 13, and interleukin 5, interleukin 4 will now act on this B cell and it will stimulate its class switching. So normally B cells produce immunoglobulin M, but when it is stimulated by interleukin 4, there will be class switching, and now it will begin to produce immunoglobulin E. Now recall that I had mentioned that T helper 2 cells also produce interleukin 13 and interleukin 5. So what is the function of interleukin 13 and interleukin 5? Always remember, interleukin 13 enhances immunoglobulin E production, and interleukin 5 has role in development and activation of eosinophils. So this was in short regarding the first sequence of event, and that was T helper 2 cell activation and immunoglobulin E production. The next sequence of event is sensitization and activation of mast cell. So what will be the process, or how will that thing be achieved? As we can see, when the B cells have been stimulated by interleukin 4, they will first class switch and begin to produce immunoglobulin E antibodies, and ultimately they will differentiate into plasma cells, and those plasma cells will secrete immunoglobulin E antibodies. So here we can see secretion of immunoglobulin E antibodies. Where will they go? They will go and bind with mast cell, because mast cells have receptor for Fc portion of immunoglobulin E, and that is known as Fc epsilon R1 receptor. And what will happen next? The immunoglobulin E coated mast cells are called sensitized because they are sensitive to the next encounter of specific antigen. So when the same antigen enters the individual, the sensitized individual, and when there is subsequent exposure to the same allergen, the multivalent antigen will bind and cross-link adjacent immunoglobulin E antibodies. So here we can see the antigen is shown in yellow color. It has bound with and cross-linked adjacent immunoglobulin E antibodies. So whenever there is cross-linking of adjacent immunoglobulin E antibodies, what will happen? The Fc epsilon R1 receptor will come in close contact, and that will result in activation of signal transduction pathway, and ultimately those signals will evoke production of mediators, and also there will be degranulation of those granules containing various mediators, and also there may be events that will cause late phase reactions, as we will see. Now let's talk about the mediators of type I hypersensitivity. So this slide is summarizing the mediators of type I hypersensitivity. As we can see, the mediators will include primary and secondary mediators. Always remember, primary mediators are preformed. That means they were already present inside the mast cell, stored inside granules. When mast cell got activated due to antigen cross-linking immunoglobulin E antibodies on mast cell surface, complex sequence of reactions will occur inside the mast cell, and that will lead to mast cell activation, and this will cause mast cell degranulation and release the primary mediators. On the other hand, secondary mediators are not preformed. They are newly synthesized after the mast cell got activated. That's why in your textbook you will see a term called de novo synthesis and release of secondary mediators. Always remember, de novo means starting from the beginning. So what are the preformed mediators? They are divided into three categories and they will include vasoactive amines, certain enzymes, and proteoglycans. Regarding vasoactive amines, always remember the most important mast cell-derived vasoactive amine is histamine. Histamine is responsible for intense smooth muscle contraction, increased mucus secretion by our bronchial, nasal, and gastric glands, and also for increased vascular permeability that we can see in type 1 hypersensitivity. Regarding the enzymes, always remember that these enzymes are seen in the granule matrix of the mast cell, and they will include neutral proteases and acid hydrolase. These enzymes can cause tissue damage and activate various components of the complement. Regarding the proteoglycans, they will include heparin and chondroitin sulfate. And what is their role? They serve to package and store the amines inside the granules of the mast cell. So always remember that the proteoglycans will include heparin and chondroitin sulfate, and their role is in packaging and storing the amines in the granules of the mast cells. So what are the secondary mediators? They will include lipid products and cytokines. Lipid mediators are actually arachidonic acid derived product, and we all know about this. When mast cells become activated due to complex sequence of reactions, that also lead to activation of an enzyme called phospholipase A2, and this enzyme can convert membrane phospholipid to arachidonic acid. And the arachidonic acid is a parent compound, and from this parent compound we get certain leukotrienes and prostaglandins. And in your exam, the examiner may ask you about the functions of various leukotrienes and prostaglandins. So let's talk about that briefly. Always remember, leukotriene C4 and D4, these are very powerful vasoactive and spasmogenic agents. So they can increase vascular permeability and they can also cause smooth muscle contraction, particularly the bronchial smooth muscle contraction. What is the role of leukotriene B4? It is a chemotactic component and it can attract neutrophils, eosinophils, and monocytes. Prostaglandin D2, its function is intense bronchospasm and it can also increase mucus secretion. Regarding the cytokines, always remember mast cells are the source of many cytokines, and I have an entire lecture on cytokines. So you can also watch that video after watching this to know more about cytokines. For this lecture, always remember that tumor necrosis factor, interleukin-1, chemokines, these are the cytokines that will promote leukocyte recruitment and they have important role in late phase reaction. So always remember TNF, that is tumor necrosis factor, interleukin-1, and chemokines can promote leukocyte recruitment, particularly during the late phase reaction of type 1 hypersensitivity. What is the function of interleukin-4? It can amplify T helper 2 response. So now that we have talked about the mediators of type 1 hypersensitivity, now let's move on and talk about the clinical aspects of type 1 hypersensitivity. Now this slide is summarizing the clinical aspects of type 1 hypersensitivity. So we can see that in the lungs, the disease that is caused by type 1 hypersensitivity is asthma. The major symptoms will be wheezing, dyspnea, and tachypnea. What is wheezing? Wheezing is a high-pitched, coarse whistling sound when certain individuals with type 1 hypersensitivity are breathing, particularly seen in bronchial asthma. So the major symptoms will include wheezing, dyspnea, and tachypnea. Dyspnea means difficulty in breathing and tachypnea is increased rate of respiration. The typical allergens for asthma will include pollen, feces of dust mite, animal danders, etc. And also certain occupational allergens may also lead to asthma. Moving on to skin, Here, type 1 hypersensitivity may result in atopic dermatitis or eczema and also in hives or urticaria. Now, what is happening in atopic dermatitis or eczema? There is itching, vesicular lesion, and the allergens are uncertain. In hives or urticaria, there is also itching, but also there will be bullous lesion, and the typical allergens are various foods and drugs. Now, in the nose and eyes, type 1 hypersensitivity can result in rhinitis, conjunctivitis, which are also known as hay fever. So there will be runny nose, redness, and itching of the eyes, and typical allergen is pollen. In the gastrointestinal tract, type 1 hypersensitivity may lead to eosinophilic esophagitis in the esophagus. So there will be vomiting, esophageal ring, and dysphagia, that is difficulty in swallowing, and typical allergens here will be various foods. Moving on to the next slide, here we can see certain systemic disease that can also result from type 1 hypersensitivity. So, anaphylaxis, the major symptoms will be shock, hypotension, wheezing, itching, urticaria, cardiac arrest, asphyxia, etc. And typical allergens will include bee venom, certain drugs, for example penicillin, and certain foods, for example peanuts. Now, the last topic that we will talk about today is regarding the late phase reaction of type 1 hypersensitivity. What do we mean by late phase reaction? There is recruitment of leukocytes that will amplify and sustain the inflammatory responses without additional exposure to triggering allergens. Eosinophils are often seen in abundance during these reactions, and they are recruited due to chemokines like eotaxins that were produced by epithelial cells, T helper 2 cells, and mast cells. So, epithelial cells, T helper 2 cells, and mast cells are producing eotaxin, and that is recruiting eosinophils. So that's why in type 1 hypersensitivity, particularly in late phase reaction, we can see abundance of eosinophils. Interleukin 5 produced by T helper 2 cells are the most potent eosinophil activating cytokine, and activated eosinophils will release proteolytic enzymes, major basic protein, and eosinophil cationic proteins, and all these things will result in tissue damage. So this is in short about late phase reaction. Always remember that here the reaction is happening without additional exposure to triggering allergens, and it is happening due to recruitment of leukocytes. They are amplifying the inflammatory response and they are causing tissue damage. So this concludes our lecture on type 1 hypersensitivity. I hope this lecture was helpful. If you like my videos, do comment, share, subscribe, and let me know. And for my students, I will also recommend you to go through your textbooks to know more. Okay, that's all for today. Until next time, take care and stay safe. Thank you.